This lecture is part of the Canadian Bioinformatics Workshop. It is released through the Creative Commons licensing agreement, and it's free to share and reuse as you see fit, as outlined in the license that is part of this presentation. Module 3, Enrichment Maps. So the next part of this talk, we've already discussed networks, we've already discussed Cytoscape, and previous to this, we've also discussed different techniques for running enrichment analysis. So how do we put all of those things together? How do we take our enrichment results and use Cytoscape and use networks to represent our enrichment results? And how we're going to do that is we're going to use a specific Cytoscape app called EnrichmentMap, as well as a bunch of other ones to take our results from the labs we did in Module 2 for gProfiler and GSCA and represent them in a more graphical representation. So by the end of this lecture, I hope that you'll understand how to transform your enrichment results from gProfiler, GSCA, or any other enrichment type algorithm to a network, understand the differences between a network, as we discussed in the previous lecture, and an enrichment map, and be able to summarize your enrichment results with annotation using Annotation app, which is another Cytoscape app that we're going to discuss. So at the end of, Module 2, we generated a whole lot of enrichment results. And we generated those enrichment results using programs such as gProfiler and GSCA. And when we actually got a chance to look at them, the result files that they created, granted GSCA creates a lot of different files, but gProfiler, for instance, just was one file that we downloaded. It was a table. We went from having a list of genes and their differential expression statistics, or just simply a list of genes as it is for gProfiler, and we translated one list into another list. And it's not so easy to interpret a list, right? Even the original list that we started with is just a list of genes. So in order to garner more information from them, we want to be able to represent this in a different format. So huge cognitive burden relating to this information. It's like overlapping pieces of information. How do we summarize it as a picture? So just basic framework of the analysis that we've done until now, right? So we had experimental data. We ran it through an enrichment test. This enrichment test also pulled a lot of information from pathway databases and created gene sets, or sets of genes that are related to each other by some functional process or pathway. And at the end of this analysis, we end up with an enrichment table, which lists all the different functions and an enrichment score or p-value associated with them. A little bit more in specifics, what did we actually do? We use two different programs, GSCA and gProfiler. GSCA is specific to a ranked gene list. It's using the entire subset of differently expressed genes, whereas gProfiler uses a subset or a thresholded list, so a selected set of genes. And you define that threshold and you define how many genes you're going to run it through. We send both of those lists through our enrichment algorithms, and we overlay them with a set of pathways that we have also collected or pulled from somewhere else. Ultimately, we end up with a set of pathways that are enriched in condition A versus condition B. So we have a set of pathways that are upregulated, and we have a set of pathways that are downregulated. Now, for GSCA, this process is done seamlessly with your one list, but in a program like gProfiler, you actually have to do a little bit more of the definition of it, meaning that if you want to get the pathways that are upregulated and the pathways that are downregulated, you have to run two separate analyses, one with a thresholded list of genes that are upregulated and one with a set of genes that is thresholded that are downregulated. And you run those separately, and then you have two separate lists that you can then use to analyze further downstream. So also, what we've done in the course is we've discussed networks, right? And we discussed that a network consists of a node, an entity, and the relationship between those two nodes is an edge. And you can define it, meaning that we've discussed protein-protein interaction networks where each node is a protein, and an edge between two nodes represents a physical protein interaction between those two proteins. And there's the concept of a DNA binding network, or a co-expression network. You define the nodes and the edges. So how are we going to go about taking the concept of a network and use it for our benefit with our enrichment type results? So now we're going to talk about a different type of network, and it's called an enrichment map. Okay, and what is an enrichment map? Here, instead of an individual node being an individual protein, each node is a pathway or a gene set. And why is this important? Why do we do this? A lot of the pathway resources that we're using contain highly redundant information. There are lots of different pathway sources that we take from, and a lot of pathways have similarities, and a lot of the data that we're getting back is a little bit redundant, right? Meaning that you have multiple pathways that do similar things, but they're all coming up as enriched. So basically, the amount of information we have coming back is actually quite a lot. And if we want to summarize it a little better, we actually want to summarize it according to theme or according to overall view, right? So now, instead of an individual node being an individual protein, an individual node is a pathway or a gene set. And the connection between those are the number of genes they have in common. So what happens is this highly redundant list of pathways that we get up or processes that we get back from an enrichment analysis, it will actually pull similar pathways together, because any pathway that shares a significant amount of genes with another pathway will be connected to each other. And then you can garner a lot more information about general themes, about general processes that are happening in the cell. Now, it's important to note that areas that are highly connected in the network don't necessarily represent a region of the network that is more important, but it specifically represents a region in space that is very well annotated. There are certain processes and pathways in the pathway databases have been studied for years and years in multiple different species and are very well represented. And then there are other pathways that only have minimal research or information about them. Now, that big massive cluster that you get in a network, now you might want to gravitate towards that and say, oh, wow, that looks really big, really, really important. It is no more important than the individual node that you might have in the network that's off on its own on the side, because it doesn't have a lot of connection or doesn't have a lot of annotation associated with it. It could be just as important. It's just a matter of a different annotation bias, I guess. So a few of the other features that we use in order to add information to our enrichment map is, so I've said already, each node is a pathway or a gene set. The size of it is by default correlated with the number of genes associated with it. You can change that size to be correlated to directly proportional to the enrichment score, for instance. So, the more enriched a gene set is, you can make it bigger. The color often indicates the class, whether or not it's up or down or class A or class B. This is only relevant, though, if we're talking about one individual comparison. In some of the other use cases that we're going to mention a little bit later, you'll see that color also can indicate data set, especially when you have data sets more than 2, 3, 4, 5, however many data sets you have. You can actually associate a color with a data set, and you can see more clearly which gene sets are associated with which data sets. Now, the thickness of the edge is related to the number of genes that two gene sets have in common. The thicker the edge, the stronger that connection is. So an enrichment map now, okay, we start with our enrichment results in our up and our down regulated set. So we have a set of gene sets that are enriched. Each node is going to be an individual gene set. And then the relationship between two gene sets is calculated using an overlap statistic. There are a few different options that you can use within the enrichment map. There's the overlap coefficient, there's the Jacquard coefficient, and there's also a combination of the two. But basically, the calculation is just looking at the number of genes that the two sets have in common and divide it by the minimum. So the Jacquard looks at the union of the two sets as being something that you divide it by, and the overlap looks at the minimum of set A or set B in order to give it a percentage, I guess, of how that overlap represents the commonalities between the two gene sets. So just briefly, what we've kind of discussed here is we start from our tabular format gene set, list of gene sets, and we take each gene set. Each gene set is a node, and the relationship it has with other nodes is the number of genes they have in common. And so we take our tabular information and we've translated it into a network. And now all of a sudden, we can get a better view of all the different pathways that are coming up and whether or not they're up or down in our analysis.